now take a look at the exact mechanism of translation. You already know that translation is a process where the information present in the mRNA, which is in the form of triplet codons, it is read by a suitable machinery, and this information is converted into a protein or a polypeptide. So basically it can be defined as a process of polymerization of amino acids into protein. So what do we require for the process of translation, or what is the machinery made up of? So for the process of translation, we need ribosomes, we need a translation unit, and we need amino acids along with the help of certain proteins, and we also require energy and magnesium ions. So when all these units are coming together, then that would help in the process of translation. So let us discuss the translation in eukaryotes, though there is not much difference between prokaryotes and eukaryotes, although there are a few differences which we will discuss when we are discussing the translation process. So first we need a translation unit, like we need a transcription unit for the process of transcription. In a translation unit, the translation unit is nothing but your single stranded mRNA molecule. It contains the start codon and the stop codon, and also the triplet codons that are supposed to be read. So this is what contains a translation unit. Also, other than this, there are few other sequences present before and after the start and stop codons. These are called as the untranslated regions or the UTRs. They also help in the process of translation. They are present before the start codon and after the start codon. So this is what is called as a translation unit. As you know, in most of the organisms, the start codon is AUG, which codes for methionine, and the stop codon could be either UAA, UAG, or UGA. Let us assume this is UGA. So out of the 64 codons, 61 codons will code for amino acid. These three do not code for any amino acid. They function as the stop codon required for stopping the process of translation. So our translation unit is ready. So this is the sequence which needs to be read and converted into a amino acid. And we need ribosomes. The ribosomes, they consist of two subunits, a small subunit and a large subunit. So what happens is, the first step, which is the initiation step, it begins with the smaller subunit of ribosome coming and binding to the start codon, that is to the mRNA. So the small subunit of ribosome comes and binds to the start codon, that is AUG. So for this binding, certain initiation factors are helpful. These are proteins which will help in this binding. They are called as initiation factors. Once the small subunit of ribosome comes and binds, it is soon followed by the large subunit. The large subunit also binds to this mRNA ribosome complex. So the large subunit will also come and bind. The large subunit, it has got three sites on it: the E site, a P site, and a site. The E site is called as the exit site, from where the tRNAs they leave. The P site is called as the peptidyl site. This is the site where the polypeptide chain is being formed or it is being elongated. And A is the amino acyl site. This is where the new amino acid carrying tRNA they come and bind. So that is the three sites EPA, read in the same direction, 5 prime to 3 prime direction. And we also require tRNA is very important. tRNA is also required for translation, which is very important. So the large unit comes and binds to the complex. Once the large subunit comes and binds to the complex, the initiation process has almost begun. Then what we need is, we need the tRNA molecule. So the tRNA molecule is nothing but the adapter molecule or transfer molecule. So a typical tRNA molecule is around 76 to 90 nucleotides in length. It is a single stranded molecule, but it will bend and coil in such a way that its structure is called as the clover leaf structure, which was proposed by Jacques Monod. The structure of tRNA. Jacques Monod proposed the clover leaf model. So the structure of tRNA, it looks like this. It has total of 4 arms or stems, and one is a variable arm. At one end we have the 3' OH group, which carries the amino acid. So let us say this is the amino acid, and another end we have the free 5' end. So this is coiled in such a way that the stems are base paired to each other, forming a strand, whereas the loops, so this is what is called, this is called as a T loop, and this arm of the tRNA is called as the anticodon arm, and in some tRNAs we have another 5th arm called as the variable arm. So everywhere in the stems the nucleotides are base paired to each other, forming a strong bond, but in the loop section the base pairing is not seen. So there is no base pair that is seen. So this is the structure of the tRNA molecule. tRNA molecule acts as an adapter molecule. So this tRNA molecule, it carries the particular amino acids and is very important in the process of translation. The anticodon arm, it contains 3 bases which can read the base present on the mRNA. So the anticodon arm will read the code present on the codon. So that is how the code is red. That is structure of the tRNA molecule. Even before initiation process starts, there is a previous process that has to take place. So the first step is charging of the tRNA or amino acylation of tRNA. It takes place. Then we have initiation followed by elongation and termination. So what happens in the first step, that is amino acylation or charging of tRNAs? The first codon is always a start codon, and the start codon in most of the organisms is AUG. In some organisms other than AUG, there could be GUG or there could be UGG. These two can also act as start codons very rarely. 97% AUG is the first codon in the mRNA process. So this AUG codon, if it has to be read, it requires a tRNA molecule. So the tRNA molecule for methionine, so this is the tRNA molecule for methionine 3' 5' and so that would be the anticodon arm will have UAC. So for the attachment of this tRNA molecule to this codon, energy is required. That is what is called as charging of tRNA. So this methionine group, it also contains a phosphate bond. When this tRNA molecule comes and attaches over here, the energy is provided by this. So the first tRNA molecule is a charged tRNA molecule. In prokaryotes, there is a bacteria. This tRNA is called as a formylated methionine tRNA. That is the difference. So the methionine is formylated in prokaryotes, whereas in eukaryotes the formyl group is not present. That is the first step, that is charging of tRNA. So now that the tRNA molecule carrying the methionine amino acid is ready, once the small unit and large unit are bound, this tRNA molecule will come and it will bind to the untranslated region. So it will come and stand somewhere over here, and they will keep on walking like this in this direction. So they don't directly come and bind to the start site. So they will keep on walking, and when the anticodon arm, it will coincide with this codon AUG, it will stop over there. So now in the first step, the tRNA anticodon arm has recognized this is the specialized type of RNA called as a charged tRNA. Anticodon arm will be able to read this code and it will stop over there. So that is the process of initiation. What follows is the second step, that is elongation. So the charged tRNA molecule carrying methionine, it will come and bind in such a way that it is present in the P site of the large subunit, and the E site is free and the A site, amino acyl site, is also free. So next what happens? In the elongation step, the second tRNA will come and bind over here. So in this case, the codon here is GAG. GAG codes for glutamic acid. So a tRNA molecule that is carrying glutamic acid. So carrying glutamic acid will come and bind to the amino acyl site. So how is this done? There are so many tRNA molecules. Each tRNA molecule will come and bind here. If the anticodon arm is not able to decipher this code, binding does not take place. So only those tRNAs that carry the particular amino acid can come and bind. So if a tRNA carrying phenylalanine wants to come and bind here, it is not possible because the binding will not be successful. So the second codon is GAG. So a glutamic acid carrying tRNA will come and bind over here, the anticodon arm. So we have this particular combination CUC. So it will come and it will bind at the A site. And now because of this, what happens is the two amino acids are present very close to each other, so there is a attraction between them. So because of the ATP molecule that they carry, what happens is a bond forms between these two. So this bond is what is called as a peptide bond. So in elongation step, peptide bond forms between the tRNA of P site and A site. So if they are close enough, it is energetically favorable to form a bond. So the bond is formed, and also once the bond is formed, the methionine group or amino acid, it is transferred to the new tRNA. So once the bond is formed, what happens is this methionine will be discharged from here and it will come and form a bond over here. So now what we have is we have a small dipeptide molecule containing two amino acids. So this methionine is now bound to this, it is bound to the A site. Next what happens is the ribosome, it always moves in the 5' to 3' direction. So this ribosomal unit will then move one step forward. So that is one codon forward. So when it moves, what happens is the ribosome moves one step forward and mRNA is going back. So now this tRNA molecule is in the exit site, this is in the P site, and the new site is now the A site is free. So the tRNA molecule which does not have the methionine, it exits, and this tRNA molecule can be recycled for carrying a new methionine amino acid. So exit site, the tRNA molecule is gone. So once this molecule is gone, as you can see, now the A site is free. So next another amino acid which is coding for phenylalanine, it comes. So we have this codon coming and binding. So UUU stands for phenylalanine. So a tRNA carrying phenylalanine comes and binds to the A site, So once again, because of the close proximity, so there is a peptide bond that is formed between this and this, and then this dipeptide molecule is transferred over here. So from this tRNA we get the chain that is transferred to this. So now you have a protein made up of 3 amino acids, and once again what happens is the ribosome unit moves forward. When it moves forward, this will exit and this will be in the P side and the A side is free. Like this, the ribosome keeps on moving forward until it encounters a stop codon. So when the stop codon comes, UGA, there is no tRNA that will code for the UGA, for the stop codon, because it does not code for anything. So when stop codon comes over here and the ribosome comes over here, along with the help of certain termination factors or release factors, so some release factors will help in this process. So the stop codon, it does not have any tRNA. So the final tRNA that comes and binds, it does not carry an amino acid. It will add a water molecule to the chain. So when the water molecule is added, the polypeptide chain is released from the ribosome. When the ribosome units will also separate. So finally what is, the ribosomes are released and the polypeptide chain is also released, the protein is also released. So that is how the information present in the mRNA in the form of codons is read by the anticodon arm of the transfer RNA, and the appropriate protein chain is formed. This protein formed it has a primary structure, it is in linear fashion. These proteins can undergo modifications, that is, they can undergo secondary structure, tertiary structure, and the proteins so formed are sent from the endoplasmic reticulum to the Golgi bodies for modifications, and from here they are transported to different parts of the cell. That is the process of translation. Thank you.